When the virus SARS-CoV-2 emerged in late 2019, it rapidly
Coronavirus
spread
officially hitting
across
the
the globe
U.S.
Cases here in the
and
U.S. have
developed
exploded.
All-time
into the
highs today in
devastating
many locations.
COVID-19 pandemic. So this was a global pandemic at that time. Hello and welcome to my channel. Today we are going to talk about an amazing topic that is about the Nobel Prize winners in physiology or medicine 2023, where we have seen that the Nobel Prize, which was given by the Nobel Assembly at the Karolinska Institute for the outstanding discoveries in physiology or medicine, to Katalin Kariko and Dr. Drew Wiseman. So they got the Nobel Prize on the 2nd October 2023 recently. Now we will see what exactly they have done. Let me give a quick introduction about the vaccines which were being used before the pandemic. So we know that the different vaccination strategies which were being used before the pandemic, different types of vaccinations were being tried. Like we have used the weakened or inactivated viruses so that we can trigger the immune response in case of, for example, in poliovirus, in measles, in the yellow fever. Also, the different vaccines based on individual viral components like the recombinant proteins in case of the hepatitis B virus or the human papilloma virus, and even the different carrier viruses as vectors have been used, like in case of the Ebola virus. So these were the different strategies for developing the different vaccines against the different viruses, and they can when they were injected into the human body, they can just to trigger the immune response. So in this way, we are able to provide the vaccination to different viral infections. Now looking into the Nobel laureates, so they basically discovered that base modified mRNA, it can be used to block activation of inflammatory reactions. So here is the unmodified mRNA. This unmodified mRNA, it can just to trigger the inflammatory response, whereas the base modified mRNA, it cannot trigger the immune inflammatory response. So the discovery of using the nucleoside base modifications in vitro produced mRNA can help us to bypass the immune checkpoints. So immune checkpoints can be bypassed and we can avoid the unnecessary inflammatory response. So this unnecessary immune response, the inflammatory response, is bypassed. Now let us see the production of mRNA vaccines. So different steps are used to produce the mRNA vaccines. So first step is the antigen selection, and then the linear DNA template synthesis occurs, and in third step the in vitro transcription will happen, and then in fourth step mRNA purification will happen. So here we can see that this first setup is the sequence design, and then here we have the in vitro transcription, then we have the purification, and at the end we can get the pure and homogeneous IVT mRNA. Now let us see the amazing part of the discovery here. The Hungarian biochemist named as the Katalin Kariko, she has an amazing interest in the mRNAs. So she was trying to see the mRNA as a therapeutic for the therapeutic purposes. So she was doing the different methods and trying to develop the different methods so that the mRNA can be used for therapy. The new colleague of Kariko at her university was the immunologist Drew Wiseman. He was interested in the dendritic cells. So these dendritic cells, they have an important function in immune surveillance. These dendritic cells, they have actually important function in immune surveillance and the activation of vaccine induced immune responses. Now let us see what exactly they are saying. The two amazing scientists who have done an incredible discovery. So what they actually are saying.
But
And we had a lot of fun.
It was a lot of years, but they weren't boring years.
It was very enjoyable. To be a scientist is a joy.
Now let us see what they are actually saying about the mRNA vaccines.
We started working on RNA to try and understand why it was so inflammatory. We were both completely open-minded. Any data that we didn't understand, and there was a lot of it, we sat down, we kept doing experiments, we kept getting results, we kept getting excited by the results.
Now here comes the breakthrough. Actually, the Carrico and Wiseman, they noticed that the dendritic cells, they recognize in vitro transcribed mRNA as a foreign substance, which leads to their activation and the release of inflammatory signaling molecules. And they actually wondered why the in vitro transcribed mRNA was recognized as foreign, while the mRNA from mammalian cells did not give the same reaction. Now let us see the further actual activation. So here we can see that these are the dendritic cells and these are the complex mRNA. So first setup is that the antigen expression in APCs, and then in second step the dendritic cell maturation and migration happens, and in the third step the activation of T cells and B cells will happen. So here the activation of B cells and T cells happens in the lymph node actually. So here is the part where they actually undergo the vaccination.
I was really happy
So
when people wrote me emails and said that they could see their parents and family members. They could meet each other.
So this was a greater part of this discovery. So let's summarize all about this. So looking into the mRNA vaccines, we can say that these are extremely safe, and these are we need less resource or the low cost, and also they are highly efficient and fast, and in future the technology can be used to deliver therapeutic proteins, and we can treat some cancer types also. And now there comes here is the part where we are saying that nowadays the era is of personalized vaccine.